Hi everyone, I'm Shaohong Feng from Zhejiang University. It's really my honor to have this opportunity to introduce this community-based and large-scale sequencing project on the bird that we have already been running for several years. We call it B10K, the Bird Consent Genomes Project. We know that the birds show diversity in many ways, and that they are good models for us to study the biodiversity. But the bird tree is one of the most challenging phylogenetic questions. Why is that? The answer can be found in the evolutionary history of the bird. The mass extinction occurred at the KBG boundary, killed off many species on the Earth, but made room for mammals and birds to rapidly diversify and evolve. New avian is a group of birds that represented more than 95 of the bird species. And the diversification of the new avian birds occurred in such rapid radiation around the KPG boundary, which means the Big Bang radiation made the relationship among the new avians largely unsolved when only using few genes. So we think that if we can have the whole genome data, can we solve this puzzle? This is one of the B10K goals. B10K is to generate the representative draft genome for the all bird. And using this genome, we can aim to infer the bird tree based on the whole genome data, and also review the links between the genomics variation and the phenotypic difference across the bird lineage, and also assess the impact of environmental changes on the biodiversity. And our project is directed by the community of Dr. Guojie Zhang, Dr. Tom Gilbert, Dr. Eric Javas, Dr. Casting Rubik, Dr. Gary Greaves, and Dr. Fuming Lei. After running for several years, we now have over 200 collaborators from all over the world, and most of our samples are from the museum. So for such a large-scale project, we are running it in 4 stages. The order level, which means we picked up one species to represent one order. The family level, again, we selected one species to represent one family, and also the genus level, and at the end, the species level. So let's first talk about the order level. The findings of the order level were published as a special issue in a Science in 2014, and we studied 48 bird genome. We did comparative genomics. We studied the evolution of the sex chromosome, and also we studied the genetics of bird tree. And of course, we use the whole genome data to infer the bird tree of life. And this is the species tree at the order level when using the genome data of the 48 birds. And you can see here that with the whole genome data, we clarify the relationship among the new avian birds. But however, the controversy, especially in the new avians, still remains. This is a paper published in Nature, and you can see that they have more bird species, but very limited numbers of loci when building the tree. And most of the loci are codings. So here you can see the difference in the new avians between these two topologies. And such difference can be caused by the numbers of species we use and caused by the different genotypes we used when inferring the trees. So in the 48 birds, we also compare the topologies inferred from the different markers when building a tree. And we have the intra-region tree and also the UCE region tree, and also the first and the second position of the extant tree, and also the third position of exon. So here, for example, we can see this group of birds, and you can see that their position on the trees changed a lot when using the different markers, which means the effect of the types of marker is very is significant when we're building the tree for the birds. So the bird tree is still the main issue when we move to the family level. So this is our family level. In 2020, we published the 363 bird genomes from the 92% of the bird family in the nature. And now we have more bird genomes, and I think we can provide a better tree for the bird, and I can also do more comparative genomics between across the different bird lineage. But at first, we needed to identify the ortholog sequence across such a large scale genomes. And usually we use the chicken reference gene alignment. But in this way, we cannot identify the lineage specific sequence. However, this lineage specific sequence are very important for us to understand the diversity of birds across the different lineage, because those new region in this lineage can attribute for their adaptability to the environment, which we are very interested in. And that's we use the reference-free alignment generated by the curators. The curators alignment can help us to get this lineage-specific sequencing. And here we plot the lineage-specific sequence identified at the most recent common ancestor for each order. And for example, this gene was required at the note of the pastoral form. But if we use the chicken as a reference, we cannot find it because the yellow box in here is just like the right one in here. We cannot identify them. And also the curators alignment provide us more loci when building the trees, which means we can have a better alignment to infer the bird tree of life. So in the family level, we infer the species tree from the region that were randomly picked up from the whole genome alignment. Because we have too many birds and too many genomes in here, the data is too huge for us to use all of them. So we just randomly pick up some. And also we infer the species tree from the different region or from the different markers on the genome, the CDS region, the intron region, and also the UCE region, because we want to qualify the difference when using the different markers at more species level. So here is a new tree we now have, and we can see the difference between the new tree and our previous tree, which means there's still some puzzle in the tree of bird life. So the paper on the phylogenetic analysis is in the preparation, and we will talk about the new tree in this paper, and also the new time tree for the bird, and also we will talk about the effect of the rapid radiation occurred at the KPG boundary on the topologies. And hopefully we can publish soon, and we can talk about more details in the future. And based on the alignment, we also did some comparative genomics on some bird lineages, like the penguin. We know that they can't fly, but they are good at swimming. So we are very interested in why they can be a good swimmer. So we started the evolutionary history of the penguins and highlighted some genes that associated with their adaptability. And also we started a relationship between the life history factors and the genomic features. We found that the body mass and the generation time were linked with the accumulation of the mutation and further influence the genome features in some bird clades in here. So this is what we did. And now where are we now for the B10K project? So far, we have already collected more than 3,000 species with the help of many museums from the world. But there's still a big gap with our goal of 10,000. So therefore, I would like to use this opportunity to call for blood samples from all of you. And we prefer the fresh tissue sample and the fresh blood sample because we can get high quality DNA from them, which can help us to get a better assembly and can do the better inference of the tree of life and also the comparative genomics. Okay, and here we use the method for the sequencing. It called as single tube LFR. This is also a code by code long DNA sequencing method. And I'm not sure if everyone knows this method. So I list the comparison between the single tube LFR and the 10x, which may be more familiar for you. And in this table, you can see that their statistical output are quite similar, but the single tube LFR is more cost efficient. So that's why we choose it, because we have too many samples needed to be sequenced. So if you have any bird samples and want to provide it for the BTNK or want to join our BTNK community, and you can, or you want to more information on these samples or genomes or anything you want to know for the project, you can send an email to Daniel, who is our BTNK coordinator for the sample collection, and he can provide you very details for the samples we need, requirement or anything you want. And at the end, I want thanks for all the museum providing us the samples and all the people who help with this project, like Peter and Daniel who help with the sample collection, and Josephine who help with the phylogenetic signal, and I work on the comparative genomics in the BTNK. And this is the BTNK website. And we are now upgrading our website, but you can still use it to search the species information and the genome information. And the new website can allow you to download the assembly and some data you want. So this is our BTNK website. Okay, thanks for listening.